Hello, so I am Natasha de Vere from the Natural History Museum of Denmark, which is part of the University of Copenhagen. I'm an associate professor and curator of botany, and I'm actually quite a new member of staff. I'm just coming to the end of my first year in post. I'm going to share with you today a little bit about what I do and my role at the museum, and also a little bit about our museum as well. So the Natural History Museum of Denmark, in some ways, is quite a new institution. So it was established in 2004 from the merger of 4 different museums of botanical gardens, but those have a much older history. So the Natural History Museum is composed of the Geological Museum, the Zoological Museum, the Botanical Museum, and the Botanical Gardens. The botanical gardens date back to 1600, and over that time there have been 4 different locations in different areas of Copenhagen. The museum collections themselves date back over 400 years, and the Natural History Museum of Denmark today is now home to over 14 million natural history specimens. Within the botanical garden, within our living collections of plants, we have over 12,000 species. But our collections and the background to our collections is actually originates in a cabinet of curiosities by this guy, Ole Worm. And he was a professor of at Copenhagen University from 1654. And this is a picture of what his cabinet of curiosities looked like in 1655. He collected items from all the way around the world and brought them together in one place to look at the amazing natural history of the world. And we still have some of his collection items today. So this item here is a gourd, which was originally in the Cabinet of Curiosities in 1655. If you look at the image down on the bottom of the screen, you can see there's like a crocodile on the right-hand side. And if you look towards the mouth of the crocodile, you can see like a gourd with a kind of a curved neck. And this is this item shown here. And the picture is me showing this item at a staff meeting show and tell. And the label on this item shows us that it was one of the original specimens within the Cabinet of Curiosities. So this is where our museum started. But we are also building a brand new natural history museum, because natural history museums and botanical gardens are so important today. It is a place where people can come to enjoy and experience the natural world, and where we can share messages of how we make a sustainable future. We can talk about what we need to do to help preserve biodiversity and the ecosystems that we are part of. And we can look at how we mitigate and deal with climate change in the future. So on this picture here, this is an artist impression. On the right-hand side, you can see part of the green areas of the botanical garden. Towards the left at the bottom is our huge, beautiful palm house, a lovely historic building full of tropical and Mediterranean plants. There's a series of old buildings which are part of our offices and collection spaces. And then at the top, you can see a huge kind of egg in the middle of the old buildings. And this is the Ocean Hall. This is a new glass-built structure, which is like a cathedral where we're going to illustrate everything to do with the oceans as they are today. And then in the middle of that area, you can see some kind of like little lumps and bumps, because actually most of the new Natural History Museum of Denmark is going to be under the ground. So under the historic buildings, under the botanical garden, there are going to be 2 floors underground, which are going to host the exhibition space for the new Natural History Museum. So one of my roles is to be lead curator of one of the 8 permanent galleries in the new exhibition building. And my gallery is all about biodiversity. We want to create a sense of wonder and joy in the natural world that inspires us to care and empowers us to act. I'm going to show you a few illustrations. These are just provisional designers' illustrations, giving you an idea of where we are. So the new museum will open in 2025, and we're in the midst of developing the new galleries right now. So you can see a little bit of the layout and some of the design and feel of what this might look like. At the core of our gallery is going to be an opportunity for dialogue space so that our visitors can connect and be able to communicate about the natural world. Some of our ideas for this is that our visitors might write a letter to the earth. This will get folded into an origami shape and released into a digital environment that they can see along with the letters of other people. We're creating a Sounds of Biodiversity exhibit. Visitors will go into a small room and they will be immersed with the sound of biodiversity, whether it's a tropical rainforest or a coral reef. They will hear what that sounds like when it is full of life and biodiversity, and then what it sounds like when that biodiversity is lost. What does an empty rainforest with a lot less birds and animals sound like, or a coral reef? And researchers today often use acoustic signals like that to be able to track biodiversity. So we'll let our visitors experience that. My particular area of research is in plant pollinator interactions. So of course, we are also featuring that within our gallery. There will be an opportunity for visitors to see plants and animals interacting on a huge wall in the top of the museum. Visitors will then be able to interact with those pollinators themselves. So they will be able to release pollinators into an ecosystem and watch the effect that has on the plant species. And at the heart of our gallery will be a representation of two of the world's biodiversity hotspots. We will feature the Coral Triangle and the Azunga Mountains. The Azunga Mountains are part of the Eastern Arc Range in Tanzania, and it's been an area of research and conservation for the museum for the last 40 years. So my colleagues have been travelling out, trying to understand the amazing biodiversity of the area and how to conserve it. And in 1 week's time, I will also be going to the Azunga Mountains for the first time to begin some botanical research and training some of our undergraduate students. And obviously at the heart of our exhibition space is our collections themselves. So it's been an incredibly exciting opportunity to go through all of the collections within the museum and look at the stories that we really want to tell our visitors. But of course, our collections are for exhibition, but they are also for preservation and for research and conservation. So part of my role is to be curator of the botanical collections, both the non-living herbaria and spirit collections, and also the living plants within the botanical garden. We have an amazing collection within our herbarium, Herbarium C for Copenhagen. Within our general herbarium, we have 1.4 million specimens, and this is plants from all the way around the world. Within our Danish herbarium, we have 250,000 species. The Greenland herbarium has 146,000 specimens. So we have a huge diversity of plants. But what's really important is those plants also take us back in time. So this is one of our oldest sets of herbarium specimens. This is a book herbarium from Greenland, which was collected in 1739. It is the oldest recorded plants from Greenland that we can see today. And this is an incredibly invaluable resource of what those plants looked like and even what their DNA was like from this period, and how is that changing as climate change changes Arctic environments. And to be able to access that material and to access the power of research from these herbarium records, we are digitizing our collections. We're digitizing all 40 million specimens, but we're also focusing on the plants within Herbarium C. And these are windows onto the past and onto our future. So our digitization project involves capturing the data from the herbarium specimens, also the images of the plants themselves. An area which I am particularly interested in is making genomic digital signatures. So actually taking DNA samples from plants from present day right into the past and using that to be able to do incredibly powerful research on the effect of climate change on plant communities. So that's some of the overview of the museum. I wanted to move now to some examples of the research that I do within the museum and how that relates to the collections and also to our visitors. So I have a new project looking at Greenland plant diversity patterns and pollination networks. So in order to conserve the biodiversity of the Arctic and to mitigate the impact of climate change, we need to understand the patterns of floral diversity and plant-pollinator interactions. We need to see what they look like now and how they may change in the future, but we can also look at the distribution and the abundance of plants in the past in order to inform what we can do to conserve them. So we can use our Greenland herbarium. So our Greenland herbarium is already digitized, and we're gradually assimilating all of that information to make it available and publicly accessible. Now, of these 146,000 herbarium specimens, there are only 766 vascular plant species, or around that number, found in Greenland. So it means for every species, we have multiple specimens, often going back in a period of time at kind of regular intervals. Now, that's really valuable for looking at how the diversity and the distribution of plants have changed. As the climate in Greenland has changed, we expect habitats to move and the distribution of plants to change. And we can also start to look at that with our herbarium specimens. The research will continue by making an open access DNA barcode reference library for the Greenlandic flora. And we'll use the herbarium material to do that using genomic skins. This is an area of research that I've been working on for a number of years now. My previous role before I came here to Copenhagen was at the National Botanic Garden of Wales. And one of the projects that I led while I was there was DNA barcoding the whole of the flora of the UK and using that for lots of different applications. We can also use machine learning with our Greenland herbarium specimens to track changes in plant traits over time. Are they flowering earlier or later? Has the distribution of stomata changed? There are many things we can look at using our herbarium records. And then the other aspect of the research that I will be doing within Greenland, but also some work in areas of Denmark and some of the other Nordic countries, is looking at plant-pollinator interactions in a bit more detail. We know that pollinators are vitally important for the pollination of crops and wild habitats and are facing increasing stresses in their environment. And it is a perfect storm of pests and diseases, loss of habitat, climate change, insecticides and agricultural intensification that cause pollinator decline in wild pollinators and ill health in managed honeybees. So key to that is a loss of flower-rich habitat for foraging. This means that if we can understand what plants are important for different pollinators, we can help to provide those within the environment. Within the Greenland project, we can say, how are these habitats changing for pollinators, and ask whether there'll be a mismatch between plants and pollinators in the future, or whether pollinators may be able to move to adapt to the changing climate. But first of all, we need a mechanism for finding out what plants pollinators visit. So it's possible to use observation for that, but you only get a little snapshot about one period of time. The work that I do instead uses pollen DNA metabarcoding to find out which flowers pollinate visit by looking at the pollen, which they use as a record so you can see where they've been. So we either collect insects and the pollen on their bodies as they've been foraging, or we can also look within honey samples and look at pollen within honey samples as a record of what the honeybees have been foraging on over a period of time. We collect the insects or the honey, we extract DNA from the pollen within those samples, and then we amplify it with two specific DNA barcode regions and sequence that using next generation sequencing. We compare it to our reference library. So our reference library for some of the previous research I've done has been the plants of the UK, and for our new work, it will be the plants of Greenland. So some examples of how I've used this research previously, and this is some work which was done in my previous job at the National Botanic Garden of Wales. So we asked questions focusing on bumblebees, honeybees, solitary bees and hoverflies. And we focus within the botanical garden, because botanical gardens are fabulous case studies and study environments where you've got natural habitats, sometimes agricultural habitats, and also, of course, fabulous gardens with an amazing array of plants. And that's a great place to look at what plants are important for pollinators in different types of environments. So we collected pollinators within the botanic garden. We looked at, do bees and hoverflies use the same floral resources? How specialized is the diet of bees and hoverflies? How does the plants they use change throughout the year? Has there been historical changes in pollinator diets? But key to the work that we did within the botanical garden, and I'll be continuing this research at the Natural History Museum of Denmark, is the societal impact that we can have from that work. So we do extensive public engagement and training programs. We talked about how do we garden for pollinators. We looked at ethical beekeeping. We developed annual seed mixes, which were based on the results of our research. And one of the projects we did, which was a nice example of how you can combine the public engagement along with the research, was a Saving Pollinators Assurance Scheme. So we worked with local nurseries who grew plants, and we used the outcomes of our research on pollinators to say to the nurseries, these are the best plants to grow for pollinators. What we then did was we looked at how those plants were grown. We did not want plants grown in peat compost or when synthetic insecticides had been used. So we worked with nurseries that produced plants organically, and then we got them to grow these particular plants. And then we had a logo which showed that if people bought these plants, we know that they would be genuinely good for pollinators. So this illustrates the type of work that we do, and I'll be continuing in my new role at the Natural History Museum of Denmark. So what I want to do to finish with is I want to show you a small film, and this really illustrates the new museum building that we're creating. And my biodiversity gallery is in the centre, and you'll see it being built with the kind of sinuous walls as we grow round. Okay, thank you very much. So that's all from me, and it's been lovely sharing with you some of the work we're doing at the Natural History Museum of Denmark.